All right. I'm going to be talking about user facilities, and let me share my screen here. Here we go. Get this into presentation mode, I hope. All right. Hopefully everybody can see my screen. I'm going to talk, kind of give a high-level overview of user facilities. And the question you're probably asking yourself after seeing John and Kristen's presentation is, where's all this cool data? How can I get some of this data myself? And is it really expensive to do so many different types of experiments? And the answer is that there are a number of user facilities available that can help generate the data that you need to accelerate your DBTL process. So what is a user facility? And I'm going to take the US Department of Energy kind of definitions because we are at a DOE in the DOE system. It's a DOE user facilities. There's some text here, and there's a website at the bottom. You can go look some of this up yourself. But suffice it to say, these are facilities that are funded specifically by the DOE to work with users, and these users can be from anywhere. We have both domestic and international users at the user facility that I'm at, the EMSL. And we are funded by DOE to work with users to generate data for research that is aligned to where DOE Office of Science wants us to go. And in our case, we work on data that supports the DOE missions in environment and energy. And I'll elaborate on that a little bit more. So what is the Environmental Molecular Sciences Lab? We are located at the Pacific Northwest National Lab in Richland, Washington. And our focus is, we're funded by the Biological and Environmental Research part of the Office of Science. So we focus on environmental and energy, in particular biotechnology for fuels and products. So this ABF, while funded by a different part of DOE, is highly relevant for EMSL's mission. We have over 200 active user projects at any one time, hundreds of users from the US and obviously worldwide, internationally. This is a US map of our users, but we do have users from all around the world. Our footprint is actually quite large, over 234,000 square feet. And our annual operating budget is currently around $43.5 million. So that is funding that comes to us to work with users to use on some of our over 150 instruments, as well as a supercomputer. So we have, and so what I'll do is I'm going to dive into the Environmental Molecular Sciences Lab user facility as an exemplar for user facilities and how we operate. Each user facility operates a little bit differently. So we're different than some of our colleagues in subtle ways, but generally we're accessed via a proposal process. And in our case, we have 3 different areas of research: environmental transformations and interactions, functional and systems biology, and computing, analytics, and modeling. And in the environmental area, we want to understand molecular transformation and transport across different scales to predict an ecosystem response. So we're interested in mechanistic and predictive understanding of environmental, microbial, plant, and ecological processes above and below ground ecosystems, and then nutrient cycling as well. it's actually a pretty broad scope. And then in the functional and systems biology area, we're interested in basically connecting phenotypes with genotypes in the areas of industrial biotechnology, so production of perhaps biomaterials, molecules that might be considered commodity chemicals, and of course biofuels. And our interests range from microbes to, or bacteria and archaea, algae, fungi, plants, and we span from structural biology all the way to understanding some of the complexity in cell signaling, how are cells in adapting to their environment from a molecular and regulatory sense. And then we have a new area that formerly we had computing integrated, and it still is integrated across the other two areas, but we decided that we really wanted to pull this out as its own science area as well. So computing, analytics, and modeling to bring advanced data analytics, visualization, computational modeling, and simulation to use on multimodal experimental data, again, to develop a predictive understanding of biological and environmental systems. So let's dive into what our capabilities are. Let's see. There we go. So we at EMSL, we've actually divided ourselves into different integrated research platforms that are groups that are grouped by science. And then within these, there's different methods that we can use to approach any sort of scientific problem. So on the biology side, we start at kind of structural biology, which is exactly what it sounds like. We're interested in atomic structures and protein molecular structures for proteins, protein complexes, protein stoichiometries within multi-protein complexes. And then as we think about integrating those proteins into pathways, we have a biomolecular pathways integrated research platform that's focused on understanding kind of how these different enzymes work in combination. What are the metabolites that they're producing? How are those metabolites transformed by the next step, say in a pathway? And then at the cellular signaling and communications level, we're interested in cellular responses to, for example, differing of these biomolecular pathways. So how does a cell respond to its environment and to different types of metabolites? How is it regulating its different genes and proteins, et cetera, in response? For our environmental IRPs, we're interested in plant interaction with the ecosystem and phenotyping of plants and how those plants respond to their environment, biochemical transformations, and then ecosystem interfaces. And then within all of these IRPs, and then we also have an integrated research platform that's in the computing area. So again, we're interested in all these different types of science, and this is actually an interactive map that you can find on our website. And so that's www.emsl.pnl.gov. You can look there. So then we have our user facility where we're working with users, but we know that we can't offer everything that a user might want. And so several years ago, in partnership with the DOE Joint Genome Institute, the FICUS program was started. And FICUS is short for Facilities Integrating Collaboration for User Science. And this allows us to partner with, say, the JGI, and in a single proposal, allow users to access both JGI and EMSL. So that's in addition to JGI has its own calls for proposals, EMSL has its own calls, but we have this combination call. And we're constantly working to work with other facilities to kind of grow the ability for research scientists to access multiple different types of resources. So this last year, in addition to having a ficus with JGI, we had a ficus with the biosands capability at the CSMB at Oak Ridge National Lab, and also with NEON for some of the environmental samples that they offer and some of their resources. So there's this opportunity to combine the power of multiple user facilities for a single research team for a single research problem. And so that really amplifies the impact that user facilities can have on user science. So as I mentioned, this was a high-level overview. You can learn more about our user program at the EMSL website. I've got the link down here in the slide. And I'll end with a kind of overview again of our 3 large science areas, and then the contact information if you're interested in learning about how to access our facilities, which is, I should mention, via peer-reviewed calls. Our next big call will be opening up in towards the end of this calendar year, early next year. It's our large-scale science, our large-scale research call, and there you're accessing EMSL for 2 years, multiple types of all of our capabilities to answer your questions. And then the ficus with JGI and other user facilities opens up at the same time. And I should mention, actually, I'll go back to this slide. There's a variety of not only experimental, but also computational resources. And so there's KBase, which will have, they're participating in our summer school and participated in last year's summer school as partners. KBase offers a variety of tools for looking at of different types of data, including genomic sequencing and transcriptomic data. So not all of the data that you generate must be generated from a user facility. You can use some of these other resources as well. And so again, around on this is a nice figure because around the outside, you've got the computational, primarily the computational and analytics projects, and then some of the more experimental bench type user facilities on the inside ring. So with that, I'm happy to take any questions. Let me leave this slide up, and I've got the chat up. Here we go. Okay, that looks like that question was answered about EMSL doing primary research. I should mention also, in addition to Leanne's answer, that a lot of, our internal research program is really aimed at developing the latest or developing cutting edge technologies that then we spin out to users. So our goal is to stay at the cutting edge of research with both our science and our capabilities. So we do have internal research, but again, there's definitely a user focus to that. What is the best way to discuss a ficus proposal with someone at EMSL? So I would say that the best way is to, depending on which topic area you're interested in, would be to reach out to Leanne, Nancy, or myself. Our contact is on the slide that I've left up in the presentation. And then also we can refer you to our colleagues at other user facilities, because that's really what the ficus is about, is collaborating across user facilities. So if it's if you're interested in genome sequencing as well, depending on what you want to sequence, we would we could connect you to our colleagues at JGI, or you can throw us all into a mass email. It's pretty easy to get a hold of us, and we're definitely always interested in bringing in new users, and bringing in more proposals. I should mention that it is proposals are peer reviewed. It's a competitive process, but we have a pretty reasonable success rate. I think this last year, our large scale acceptance rate was somewhere between 25 and 30%. More questions? Can you describe changes recently made to the ficus proposal process? Relative to, so we are the process changes in small ways every year. I can just talk about our current process. Our current process is that we ask initially for a letter of intent, so we can make sure that before a full proposal is written by the proposers, that it is aligned to our mission and the call. So we're very interactive. We can help users make sure that at the LOI process there is alignment. It undergoes peer review, and then we allocate our resources. So I don't know if that answers the question about changes. Off the top of my head, I can't remember what tweaks we made this year, but we are all about making sure that the proposal process is not overly onerous, and that proposals are submitted correctly. So please reach out to us and we can help with any stage of that proposal process. Is there a best way to discuss a ficus proposal with someone at EMSL? I think I answered that. Is there a particular recommended route for collaborations with user facilities to support ongoing DOE projects beyond general public calls like ficus? I think the best way to collaborate with any of the user facilities is to contact the scientists at the user facility. We can collaborate directly with on projects, at least within the US. We have mechanisms for that. I don't want to go too much into the weeds on that, but again, best way is to contact any of us on this list, and if we're not the person, we can quickly find that person. Would you like to share your bigger objectives with us? Sure, happy to share the bigger objectives. Right now at EMSL, for example, we've developed a strategic roadmap, and I'll happily talk about our long-term objective within functional and systems biology is to develop a digital phenome platform where a user comes to us and maybe they want to understand the behavior of their organism or predict what a phenotype might be based on potential changes, that they might make at the genetic level through some sort of genetic engineering. And we hope to offer a suite, or we now offer a suite of tools that allows some of that, but we're intentionally moving towards, increasing our capabilities to go after understanding proteins of, for example, of unknown function, and integrating more modeling. And you'll see some of that during this week about talking about how multiomics data integrates into models, and that those models and simulation tools might be used to predict phenotype. A lot of kind of our long-term strategy shows up on the website, so you can find detailed documentation of the directions that we're headed. on our website. I hope that answers the question. There are longer term goals also in the environmental transformations and interactions, and that's focused on kind of environmental, developing data from a wide variety of environments across the U.S. and in the world. For example, characterizing dissolved organic matter in different types of environmental samples, so that we can bring that data together and better understand or predict what's going to happen in the environment. And then in computing and analytics and modeling, we're again developing a wide variety of tools. And Leanne, do you want to pop in and talk about, since this is your area, the computing, analytics, and modeling long-term goals?
I could if I'd been listening. I'm sorry, I was responding to a comment in chat and I was like tuning out to what you were saying, actually.
Oh, wow, that's awesome.
I tried to tackle questions in real time with both of us, and now I'm failing to like keep up with what you're talking about.
That's okay. I was gonna say, what is the in a sentence or two, what's the bigger objective in computing, analytics, and modeling, so that decadal goal?
Yeah, so we do have a big focus on really pushing analytics and modeling, pushing the needle further than where it is today, right? We really look at a lot of correlations in these large scale data, but we have the goal of moving past correlating what we observe and to really being predictive. So I think in a word, if I had to put a word on our longer term objectives, it is about being predictive and not correlating what we see in the past. Yeah, and at a simple high level statement, that's it.
Awesome. All right, thanks. Sorry for putting you on the spot.
I'm looking at the question in a Zoom chat about institution where there are limited facilities and developing a proposal. I'll finish this out, but really our recommended path to being successful in a user proposal is working with us ahead of time, right? We can really help you craft making that relevance to DOE mission and what might be successful, right? We've been doing peer reviews of proposals for many years now, and we can really help you craft something that would be successful in peer review. So that's our strongest recommendation to users routinely, is not to write a proposal separately and then heave it over a wall, but chat with someone ahead of time, because we really can help you.
Yeah, and I'll add that it's probably worth delving in, and I can post some, I'll post some links after I'm done answering questions. There are programs where we can collaborate with researchers, not only through our user program, but if there is a user proposal or a project in place, we can support visits of graduate students or postdocs or faculty members to EMSL. I'll post some useful DOE programs for graduate students and faculty in the Discord chat after we're done. Are there any programs similar to FICUS for biomedical applications which do not seem to be under EMSL's umbrella? I don't have any detailed knowledge. I think NIH does support some kind, not necessarily, well, they don't support EMSL, but they do support kind of facilities. The one that I can think of off the top of my head, there is the Pacific Northwest Cryo-EM Center, is an NIH program that'll do structural biology, and actually PNNL is part of that, but that's run out of the Oregon Health and Science University. So I think there's 3 big cryo-EM centers for example, that will work with users to do structural biology. I'll try to post some links after this to at least PNCC, because that one I know about.
Yeah, I think that there have been NIH centers for various types of omics as well, dispersed, right? There might be a
Yeah.
center for proteomics in one place and a center that does sequencing in another. I don't know that there was ever a coordinated way to propose across some of those centers, and I'm not sure how long they're funded for.
Yeah, that's a great point. I'll put the information that I about for the one I know about PNCC. I'll put that in the web. How many are on the Discord chat? How about international projects that are primary in UK? I don't know off the top of my head. We have had some. I can think of some past ones that have come out of the UK, in particular about white rot. I think it was white rot or brown rot fungi. I think it was white rot. And so we do have international users. So if you're interested, again, contact us. We want to work with anybody that's got problems that are relevant to our mission. They don't have to come out of the US. What are some of the other learning workshop avenues that are available for beginners interested to know more about computational modeling and analysis aspects of omics data? So there are, we have this summer school and we're having public lectures all week and then some in-depth tutorials for a smaller group. But there are webinars. EMSL is just standing up its own Learn site, so where we'll be trying to accumulate a lot of the summer school materials, but also webinars. We plan to have webinars throughout the year, and I know that the Joint Genome Institute has a series of webinars that they've posted from their site, and we've collaborated with them on joint webinars. So there are opportunities throughout the year. Hopefully, if you signed up for this, I mean, EMSL does send out blast emails, so check our site regularly for schedules of potential webinars. Any other questions? Did I miss any?
Not seeing anything else.
All right, well, I think it is a good time for us to, I'll stop sharing, and it will be, it's probably a good time to take our break, which I believe we are reconvening.
Yeah, we're reconvene at 11 o'clock Pacific time. So in about, 32 minutes.
Okay, thank you. I was having trouble getting my screens cleared so I could find the agenda again. Woohoo! So it's a good tag team. All right, so thanks everybody for the questions. Please feel free to keep posting them. And we'll see you all back here in half an hour.